Everybody, today we are going to go over the first part of protein synthesis. Basically, we're going to learn how does DNA code for the proteins that make up our bodies. So from gene to trait, from DNA to the things that appear like our hair color, our eye color, our height, whatever, how does it happen? So first of all, we want to go over the central dogma. I know we went over this before, but DNA is transcribed into RNA. You see that in the picture. Here is transcription, DNA to RNA. You can ignore the backwards arrow. And then RNA is taken, and it can be turned into a protein, or it can be helped used to make a protein. So this is a central dogma that DNA copies itself into RNA because RNA is easy to use, it's expendable, and then we can use that RNA to make a protein. So here we see the same thing as before. We have DNA here. DNA, through the process of transcription, which you see in this box, is making RNA, and RNA is then going to be used to make a protein. And that is the central dogma. That's how information flows in our bodies. So first of all, what is RNA? We went over this yesterday, but just to review and to make sure you have it in your notes, RNA is single-stranded, whereas DNA is double-stranded. RNA contains the base uracil instead of thymine. Thymine is what DNA has, but RNA doesn't have thymine, it just has uracil. The other bases are the same, the A and the C and the G. That is not different. The only thing that's different is the T. The sugar in RNA is called ribose, hence the R, and DNA has deoxyribose, hence the D. There are also three types of RNA. There is messenger RNA, there is transfer RNA, and there is ribosomal RNA. We're going to get into each of these in more detail. Messenger RNA is going to be pretty important. This is the copy of DNA. So notice I'm highlighting copying of the DNA. And the messenger RNA is going to take the code of DNA and it's going to take it to the ribosome and allows the ribosome to make proteins. All right, the transfer RNA is going to carry the correct amino acid to the ribosome. We're going to talk much more about that later. And then ribosomal RNA, or rRNA, that's the stuff that makes up the ribosome. The ribosome is made up of RNA, a specific type called rRNA. So what is RNA? How is it different than DNA? RNA is usually shorter, right? Usually we make a copy of just one gene at a time and not the entire DNA strand, just one gene on the DNA. RNA is found in the nucleus and the cytoplasm, whereas DNA is found only in the nucleus. We can see that DNA is double-stranded, whereas RNA is single. DNA, deoxyribose, RNA, ribose. And then the bases are different. Notice the one difference, thymine versus uracil. All right. Okay, so the DNA of a person contains about 75,000 genes, and these genes code for proteins. Whenever the body or cell needs more protein, it has to use this process that we're going to learn over the next few days. So for example, your hair is protein, and if you grow more hair, that means you have to make more keratin, which is the protein that makes up your hair. When you need to make more red blood cells, there's a protein in your red blood cells called hemoglobin. Your body needs to go through this process to make hemoglobin. If you want to digest milk, you need lactase to process the lactose in the milk. So everything that your body does is based on proteins. Okay, we are going to talk about the first step of protein synthesis. Step one, there's two of them. Step one is called transcription. Transcription happens in the nucleus. This is the location. Transcription is when we copy the DNA into mRNA. Remember what the M stood for? The M stands for messenger RNA. Only one strand of the DNA is read. You can see that in the picture, right? How only that bottom strand of DNA is actually being read. And the enzyme that's important here is RNA polymerase. Remember that it's not DNA polymerase, it's RNA polymerase. RNA polymerase is going to initiate the making of mRNA. In this picture, the mRNA is red. So how does it start? Transcription begins before a gene, so right before a gene, and starts on the DNA site called the promoter. The RNA polymerase binds to the DNA and unwinds it. So let's take a look at this picture and find out what's going on. So over here on the DNA, this is the promoter. This is where the DNA sequence tells RNA polymerase, this big orangish blob, this enzyme, is going to bind back here at the promoter. And what RNA polymerase does, it does two important things. Number one, it unwinds the DNA. So notice how that DNA gets unwound. And then it's going to put in the correct bases of RNA. So it's going to put in the correct RNA bases and match them with the DNA. As the RNA polymerase moves down the DNA closes back up. See how it's closing back up on the left? And the RNA that is created is basically going to float away from the DNA. So here we see a picture of the RNA polymerase binding to the DNA. So the RNA polymerase might be hard to see. It's this part. All right. So now the RNA polymerase has bound. It's unwound the DNA. And so now what it's doing is it's adding in bases. So you can see that happening here. Bases being added. We're going to add a base here, and there, and there, and there. We're going to see a bunch of new RNA bases that are being added in. And as the polymerase moves to the right, we are going to see more unwinding, and we are going to see more bases being added to make this RNA strand in red longer and longer and longer. Here we see that happening. Notice that it's the correct base pairing. The T bonded with the A in RNA. The DNA is A, the RNA is U, DNA is G, the RNA is C. Here the DNA is T, the RNA is A. And notice here the DNA is A and the RNA is U. So we are unwinding and binding the correct RNA nucleotides. The RNA polymerase is going to continue to add bases until it reaches the sequence of DNA that tells it to stop, to hold on, we're done, transcription is over, the RNA polymerase breaks off, and the RNA gets modified before it exits the nucleus. We'll talk about that later. So here we see it all falling apart. The RNA is becoming detached, and the RNA polymerase is also detaching. And there it is, all done. Here's the RNA molecule. Notice that it is single-stranded. Notice that it's shorter than the DNA. And notice that it would, if you could tell, it would have uracil instead of thymine. And that is transcription. So we see here, we have DNA on top, and we have mRNA on the bottom that matches up correctly with that sequence. The DNA was TAC, the mRNA is AUG. So, to review, RNA polymerase binds to DNA at the promoter, and that's going to unwind the DNA. Step 2, RNA polymerase moves down the DNA strand when transcribing it until it reaches the end. And then the newly made mRNA breaks away, leaving the nucleus. The RNA polymerase transcribes the following DNA sequence. What is the sequence in mRNA? This is a good question to answer. Here is our DNA sequence. Line it up with the appropriate mRNA now. Right, of course, the correct answer should be T matches with A matches with U, if it's mRNA, G, U, C, A, U, C, A, G, C, U, G, G, A. And that is the correct sequence. And that is it for today. That is transcription. We're going to review this when I see you next. So let me know if you have any questions.